We are live.
Wow.
Okay. Well, hello everyone. Um, Quick disclaimer before we get started. The information offered in this program by the host and guest is intended for educational purposes only and does not constitute medical advice from the Mount Sinai Health System or the PolyBio Research Foundation. Now that we've gotten that out the way, I'd love to introduce myself as my name is Raven Baxter. I am the Director of Science Communication for the Cohen Center for Recovery from Complex Chronic Illness, also known as CoRE. And today we're having our first core knowledge session, which I'm very excited about because I find this topic of pathogen persistence entirely fascinating. There's a lot of interesting science behind it. And who better than to discuss this with than Amy Proal? So, Amy, would you like to introduce yourself and David as well?
Sure, yeah. I'm Amy Proal. I'm a microbiologist, and I'm also the president of PolyBio Research Foundation. We're a nonprofit organization. We're founded about 5 years ago to study what we call infection-associated chronic disease, which are conditions initiated or exacerbated by infection. In fact, most major pathogens, whether they're viral, like dengue or Zika, Ebola, or whether they're bacterial, like Borrelia, the Lyme pathogen, Bartonella, Q fever, are associated with a syndrome in which a percentage of patients do not recover and they develop chronic symptoms that can last a lifetime and be incredibly debilitating. And we want to understand why that happens, and we want to get people better. So that's our mission. And as part of that, I met David, and now I spend part of my time in addition to that as the scientific director of CoRE, which I love doing because what I do is we take all the knowledge we're gaining from the research that's happening in PolyBio, and then I'm able to take that and try to apply it to really actually trying to create a model for patients to get better. So that's what we're working on. And as part of this, the trend of pathogen persistence in these conditions initiated or exacerbated by infection is a big one, and one I really look forward to talking about more today.
Wonderful. Thanks, Amy. And David, can you please introduce yourself to everyone?
Yeah, absolutely. David Putrino. I am a professor in the Department of Rehabilitation and Human Performance at the Icahn School of Medicine at Mount Sinai. And I'm the Nash Family Director of the Cohen Center for Recovery from Complex Chronic Illness, or the CoRE. I hope from all of these new knowledge sessions that we do, we can just call it the CoRE from here on out because it's a mouthful. But, you know, the CoRE is the fifth center that we have built at Mount Sinai in just over 7 years. And in my role of rehab innovation, you know, I was tasked with a very specific goal, which was if there is something out there that is, you know, able to help patients that we're not getting out to patients fast enough, it's my job to accelerate, because the national average is that it takes 17 years to go from bench to bedside with an actionable intervention. That's an obscene number. I want to be really clear on that. And when we started our mission with rehab innovation 7 years ago, we vowed to really work every angle that we could to make that number smaller, and to make sure that actionable treatments could be put into the hands of patients first. And since then, we've successfully opened 5 different centers, all with a different clinical focus. And the CoRE is our latest, and it's all about post-acute infection syndromes. And, you know, as we started to dig deeper and deeper and educate ourselves on these, persistent pathogens were one clear pathway by which people remained sick after an initial acute infection. And so a lot of our efforts are going toward understanding how these pathogens persist and then how to treat them.
Thank you, everyone. Yes, and I'd love to dig a little deeper into what the relationship is between CoRE and PolyBio before we get into the pathogen persistence.
Yeah, it's pretty straightforward. What David alluded to, where he's been foundational in moving things more quickly than usual in spaces in which that needs to happen. I noticed that really quickly. I think there was almost a journalist asking David and I when we first met, and it was towards the beginning of COVID. And
I think August of 2020.
I had maybe just written, along with my colleague at PolyBio, Mike Van Elziger, a paper on biological mechanisms that could underlie long COVID. Someone put us in touch with David's team, and they were willing to Zoom right away. Just got on the Zoom, this whole team lined up, and they were like, tell us what we should be doing, right? And this is what was so cool, is instead of saying, here's already our, our pet theory or what you should, they just already deferred to Mike and I, who kind of been in this space a little bit before, and asked us our thoughts and really ran through all these actionable possibilities. And from that point on, I started to realize that working with David was going to be a way to, like I said before, take a lot of intellectual knowledge and be able to move it into a space where it can really help people. So as PolyBio, we just started working together. And what I think I do at CoRE is honestly just, I'm on top of the big picture of the vision of how things go, right? And since I've been in this space of infection driving chronic diseases for over a decade, I know, kind of what's worked, what hasn't, who's doing what, across the space. And I bring that down, including a lot of the knowledge I have on infection as a microbiologist, and just oversee in a big picture capacity to make sure that the studies and the trials we're doing are as cohesive as possible and are really following the biggest leads.
And I would just 100% second that, and just say that, again, this has been our process for a really long time in all of our centers. Is we don't much care where the good ideas come from. What we want to do is we want to hear from the smartest people in the room. We want to hear from the subject matter experts who are driving the basic science. And then we want to scale and implement the outcomes of that basic science. So, having PolyBio pulling together having PolyBio, first of all, being led by some of the finest minds in the space, but then pulling together some of the researchers around the globe who are doing the most important work, and having the opportunity on our end to say, okay, what's the so what in all of this research? How do we rapidly implement it into the clinic? And what does that mean from a perspective of talking to the FDA? What does it mean from a perspective of talking to CMS so that we can bill insurance? So these aren't just cures or treatments that are available to people who can pay $10,000 a pop. How do we make this scalable and actionable for a growing population of people who need it?
Thank you. Okay, so let's segue into some of the areas of focus that we have in our research that relates to pathogen persistence.
Where do you want to start?
Yeah. So what are we doing at CoRE too in relation to pathogen persistence?
Yeah. Amy, do you want to take it for some of our lead projects?
I think I'll give some background to. So obviously, when you have a condition where someone gets an infection like SARS-CoV-2 and they get COVID and they don't get better, it's a low-hanging fruit in a sense, and sort of obvious to at least pursue the possibility that maybe they have symptoms because they didn't fully clear the pathogen, right? And so the thing is, it sounds very straightforward to study and do, but there are some caveats, because as a general rule, when pathogens persist over time, they don't persist in the blood, or sometimes even in saliva or other accessible fluids that are the most commonly collected and analyzed, especially in a doctor's office. Being in the blood is actually kind of not the smartest place to be. That's where the immune system can often be the most robust, so they're going to get targeted. So in simple terms, they move and they hide in tissue or in other body sites like nerves, where they're sometimes better protected from the immune response. And that's what we mean when we say a pathogen reservoir. So like a small amount of pathogen that's hiding out, right? So the thing is, people can harbor these reservoirs after an infection, but it takes, special types of studies to find them, right? And the other thing is that the person will look like they don't have the pathogen on regular blood work, right? So that is confusing and has led to confusion in this space. So I think part of what we're doing at CoRE is that the biggest trend for pathogen reservoir is to collect tissue samples from patients in addition to just fluids. And for example, we're doing that with a collaborator, Saurabh Mahandru. We're working on a study with him where he's helping certain long COVID patients who qualified to undergo a colonoscopy procedure where safely some extra tissues obtained from that procedure that we can then analyze and look with his team for the SARS-CoV-2 virus in that tissue where it would be expected to persist, right? So moving into studies of tissue is one of the things that we're doing. And from there, David, I don't know what else.
I'm really excited about the work that we're doing with Ed Breichwitz as well. This idea that a lot of people with post-acute infection syndromes, including long COVID, experience either an activation or a reactivation of persistent tick and vector-borne pathogens. So, just starting to do some of the foundational work to understand, okay, you have long COVID, but how many of this population with long COVID have long COVID plus evidence of a tick or vector-borne illness? How many people with ME/CFS have other enteroviruses, affecting their bodies, but also tick and vector-borne illnesses? A lot of these persistent pathogens, there's a lot of, good faith basis evidence to think that some of these persistent pathogens may roll in packs. Some of them may be more pathological or more damaging to body systems if there's more than one pathogen present in the body, and they feed off one another. So, as Amy mentioned, a lot of the work that we're doing right this minute to study persistent pathogens, it is really some of the foundational work, which, I hate to oversimplify, but there's some of these questions that, as Amy mentioned, they're obvious questions. These are obvious questions that we should have been looking into quite some time ago. And we're really, passionate about searching for answers now.
Yeah, exactly. And what David brought up with what I call multiple hits is important in the way that we look at these cases. I think that, you could, when you get a SARS-CoV-2 infection, perhaps the SARS-CoV-2 virus could not fully clear and might persist in a site like the gut, or for example, the lungs, or even the lymph nodes, right? But another trend that we have found seems to be occurring in some patients that we're also studying is that the infection, the acute infection, can sink the immune response in a way that could allow other dormant pathogens to reactivate and themselves also potentially drive chronic symptoms. So for example, we see some signs of Epstein-Barr virus reactivation in certain patients with long COVID. That's especially in some work that's done with Akiko Iwasaki at Yale, who's one of our collaborators who's doing really deep profiling on the herpes viruses and even human endogenous retroviruses, which are these viral elements that can incorporate into the human genome, but also kind of have pathogenic properties, as with her lab on several of our CoRE participants as part of a study. And to clarify what David was saying before is that, and especially in the spirit of CoRE being the Cohen Center for Recovery from Complex Chronic Illness, the Cohen Foundation is passionate about better understanding the degree to which tick-borne and vector-borne illness can just be one of the contributors also to the chronic symptoms that people develop after infection. And there are tick-borne pathogens, Borrelia, the Lyme pathogen, but also other vector-borne pathogens like Bartonella and Babesia. And those pathogens can sometimes also sort of not drive symptoms in people, but potentially infection with SARS-CoV-2 or another infection might also sort of activate them and allow them to drive more symptoms, similar to Epstein-Barr virus. So we're actually looking at this wider spectrum of both viral and bacterial pathogens in their capacity to sort of, change their activity, often from like a latent type state to a more active state in patients as just, again, one of the drivers of things that we're looking at in these patients.
Thank you. So I want to naturally walk this back a little bit and get to the basics of persistent pathogens, because I did put out on the CoRE socials gauging people's prior knowledge. Do you know what a persistent pathogen is? And it was about split 50-50, where half of our audience did know and half didn't. So that really does mean that we have to go back to explaining, what is a pathogen? What does it mean to be a persistent pathogen? And kind of walk from there. So this has been really interesting for me to learn. As a molecular biologist, I'm always thinking of like things that are very small and happening inside of our cells. But it's very fascinating how things that cause disease in the body may or may not be even detected, by our bodies, because they might be really good at hiding out in different places that are outside of our bloodstream, where a lot of those, white blood cells and whatnot are looking for things. So, with pathogen being a disease causing organism, like let's define that, and persistent meaning going on, existing for long periods of time. Persistent pathogens is referring to disease causing organisms that are existing for long periods of time. And the issue is that when our body doesn't clear these pathogens, and when those pathogens have the ability to hide from the immune system, they can cause long-term issues that persist over long periods of time. And so the issues can be that we can't necessarily use blood testing to find these persistent pathogens. To find them, we may have to sample tissue. And Amy, is it true that even if we think that the pathogens are hiding in the tissue, we wouldn't even necessarily know what tissue to look at?
No, I mean, we have clues on what tissue types to look at, and I think that one thing is that the pathogens sometimes might be in the blood. It's just an unreliable location. Like in an analogy, if you think about it, if you're looking for a criminal that's hiding out, right, in a city or something like that, the equivalent of what we do in a lot of science and all of clinical medicine when it comes to persistent pathogens to a large degree right now is it's like we go into the street and we see if like we would be looking for the person like out in the open, right? And okay, that why would they be there really? Like that's a cursory attempt. You would have to go find them, and to find them, not only do you need to try to find the location that they're in, like let's say the gut, but you also need to use tools that like advanced technologies that are going to maximize your chance of finding them too. And that's another thing that's important because, some of the things we're experimenting with at CoRE are, for example, like we said, we want to know, for example, if maybe someone might have continued to harbor SARS-CoV-2 in the tissue of their guts. But what if other bacterial or viral organisms could also be in that same location as the SARS-CoV-2 virus, potentially, and that could also be a driver of symptoms? Then you want to be able to have technologies then that could find all of the bacterial or viral or even fungal organisms in that tissue sample. So part of what we are doing is we've begun collaborations, for example, with a group at the J. Craig Venner Institute in San Diego, where we're working on developing technologies that can pull the genetic material out of a sample and potentially assign it to any kind of bacterial, fungal, or viral organism so that we could have a more comprehensive potential, future biomarker of persistence. So it's basically a matter of continuing to look in sites like the gut or even the lymph nodes, which are common sites where organisms and pathogens hide out in a persistent state, and continuing to modernize the technologies and the way that we find them to maximize our chances of the approaches working.
I think one of the fascinating things that we've talked about, like in the conversations that you and I have had, have been just the notions of our bodies being sterile and how that may be a misrepresentation of how our bodies actually function and what's inside of us. Can we talk a little bit about that?
Yeah, I think, a lot of why pathogen persistence hasn't necessarily been pursued as much as it could have in this field of the post-acute infectious syndromes or infection-associated chronic disease, whatever you want to call it, is because there has in the last decades been an over assumption that the human body just doesn't have any organisms in it at all. Like, in other words, whenever teams would find signs of inflammation or problems in the body, they would just assume the immune system itself had broken down. It's kind of like a basis for primitive autoimmunity, or let's say mitochondrial dysfunction. They would just assume there has to be something inherently wrong with the mitochondria, right? And then around the year 2010, which is when I first got really inspired actually to become a microbiologist myself, teams started to take new tools that could go beyond kind of conventional, like old school laboratory culture methods to find organisms in the human body, and actually use computer tools to try to find them. And basically what happens is a team takes a sample and can understand, like knows the backbone signatures of different organisms or pathogens in the body, and can start to match those using computers. And that just blew open what we understand about the human body, because we started to use these tools on all kinds of different human sample types, and we realized that there are trillions of organisms in the human body. That's the human microbiome. In other words, we're seeded with bacterial organisms, fungal organisms, archaea, communities and ecosystems of these organisms, really as robust as a rainforest, honestly, in terms of just all the organisms and the interactions. There's even viruses called bacteriophage that infect the bacteria and modulate their activity in these communities. And there are millions of bacteria, but there are 10 times more of these phage, right? So there are quadrillions. So we have these just incredible rainforest-like ecosystems in us. And that paradigm shift has taken time to infiltrate the way we think about human chronic disease, because it changes everything. When you see now inflammation, or for example, mitochondrial dysfunction in a patient, you can say, like, wait a second, are any of these organisms, like, alone or collectively underlying that inflammation or responsible for the root cause of driving it? Same with something like mitochondrial dysfunction. Every organism has to modulate mitochondrial activity in order to replicate or survive. So there's a constant interplay between organisms and our mitochondria. So all of a sudden, all of these questions about chronic disease are approached in a new light. And that opens up then the whole field of pathogen persistence of pathogens like SARS-CoV-2, or say something like Borrelia, becomes a lot easier to put into a framework in a context where we understand that the human body is not sterile, and in fact does host all of these organisms, right? So yeah, it's a very important thing to always be thinking about.
That's really profound. And it just makes me think of how diverse even the field of biology is. And when you think about all of the things that ecologists study, like on that greater, like in the animal community, and how all of these species interactions play out in the wild and stuff. And then you think about that happening in our bodies, and how you just described this being a new a new place in the field is really incredible. And it just shows it's really promising, because we do have the tools to really push our knowledge to where it should be so we can get answers. But mind blowing when you think about how much now we don't know, because it's all so new. Wow. Yeah, David, I don't know. Does this blow your mind too?
Constantly blown. I mean, anytime you let Amy go, your mind is going to be blown. And to pull it back in the direction of clinical implementation, I think this is why we get so fired up as a team for new research that needs to push new boundaries and new frontiers, because there is all of this discovery occurring, but in many ways, it's not being researched or implemented, with respect to new conditions or old conditions nearly fast enough. And, Amy had the sort of analogy of a burglar and trying to hunt down a robber in a city. I always go toward the analogy of, the person looking for his keys under the street lamp, and someone walks along and says, why are you just looking under the street lamp? And the guy says, well, this is where the light is, and that's how medicine functions, and that's how medicine progresses. It's like, wait, what? There's all of these pathogens in tissue, but we're just testing blood, or we're just doing this. Oh, but this is what we know how to do. This is how I know to get, Quest Diagnostics to, cover your tests with insurance, and it's not congruent with what the science is doing, which is why, this is why when we're seeing all of these possibilities where we could be sprinting ahead with research, and we're also seeing in the same breath people wanting to research cognitive behavioral therapy and exercise for these conditions, this is why we raise our voices. This is why we get fired up, because there is so much complexity happening. There is so much really important science that is on the bleeding edge of solving these issues. We can't be wasting money on interventions that we know haven't worked for 30 or 40 years if you would sit down and speak with a patient. And let me give you the red hot tip. We know that they're not working, not because, maybe we should have done high intensity interval training rather than steady state training on a treadmill for 5 minutes. That's not the difference. That's not the thing that is going to make someone, be bedbound, suddenly not be bedbound. This is the work. This is the work that's going to help us to understand what is happening in the body and how to treat it.
So I'm reading a question from the audience, and really they're looking for if blood tests, like what use are blood tests to patients who might suspect persistent pathogens? And then if a blood test may not be useful, what are other things that providers can look at to answer some questions in the realm of persistent pathogens?
Yeah, I mean, that's a great question. It's like the $1 million question in this space too, because the thing is, we want to be able, to the extent possible, to be able to collect blood and saliva, accessible fluids, because that allows for the most accessible research. It sometimes means that we could collect samples from people who are housebound or bedbound, where we could, get blood and we can actually bring it. We actually do that. We have a grant from Uber where we go now to the homes of bedbound ME/CFS patients and we get their samples there because they're too sick to come in, right? So in those senses, you always want that accessible biomarker, and you don't really want to have to go into tissue if you don't have to. And I think key is that there have been, let's just take long COVID and SARS-CoV-2 reservoir as an example here. There have been some teams that have found the RNA of the virus in long COVID blood actually over periods of time. So for example, one team actually did a digital transcriptomics. It's a complicated technology, honestly, but they found transcripts from the SARS-CoV-2 virus. In fact, some that could indicate replication in blood from long COVID patients. So sometimes the RNA could be there, and we're trying to replicate that work and see, but it worries me to count too much on RNA being in blood, especially over long periods of time, because like I said, it's really over time that the pathogens tend to move and hide and get deeper into the tissues and the reservoirs, right? So in this way, relying on finding the genetic material in blood might make us miss the people who have the deepest reservoirs, who can sometimes be the sickest. So it's just a consideration to keep in mind. That being said, one of we're still looking at RNA in blood, though. It's not over. It's an open question. But one of the other leads in this space is that teams, and this includes David Walt's team at Harvard, are finding small amounts of SARS-CoV-2 protein, like spike, in the blood of some long COVID patients. And the reason they're able to find that is because they're using really what's called sensitive tools. And what I mean by sensitive is that they can find tiny, 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 tiny amounts of protein, right? Minuscule. And that could be possible because what we think might be happening is that maybe someone is harboring, let's say, the virus in their gut over time. But that reservoir is causing the lining of the gut to become worn down, inflamed, like leaky gut in a sense, right? And then maybe that allows viral protein, like spike, a little bit to leak into the blood where it can be measured, as long as you have that tool that can find the tiny amounts. And that's where the technology innovation is so important in this space. And so, for example, when we work with the Kiko Iwasaki's team, they are working with a group that has a similarly sensitive assay. So it's all about like trying to understand where the pathogen might be and continuing to up the technology. So I'm actually, the way I see it in the coming years is that we might be able to get sensitive tests that can find sometimes persistent pathogen genetic material or tiny amounts of protein in blood or saliva. But I still think that it would be paradigm shifting. And David, we wrote an application to ARPA-H, I'll be honest, for this, to see if sometimes maybe a routine biopsy, like a lymph node biopsy, could be performed on a patient with long COVID or a related condition, because that's honestly where we would most expect to find some of these persistent pathogen genetic material. And in conditions like cancer, or that we take very seriously, it's no big deal to bring in someone for a biopsy. We do it all the time. When we think that the information from that biopsy is going to really matter for diagnosis or treatment, insurance comes in and covers a biopsy. So I do think that part of what we're doing too is enough research to perhaps set a precedent where patients with post-acute infectious syndromes could actually merit getting biopsies as part of their clinical assessments. And that's something that we want to try to take on more at CoRE, depending on what we're able to pull off.
Couldn't agree more, and I think the cancer analogy is really apt. It's you know, no one's sort of there's not an oncologist in the world that sort of went, oh, you know, this is getting too complicated. We're not going to go deeper and look for answers in the organ being affected, you know, and so similarly with these conditions, I think that is the sort of area that we need to really emulate. Is this is a complex chronic illness. We need to be able to look closely at the at the organ that we think is affected, or at the organ where we know this particular pathogen is most likely to be harbored. And here's our precedent. You can't test someone's blood. This has been a holy grail of cancer, by the way, is the ability to test someone's blood and say, you have melanoma, you have this, you have that. They haven't gotten there yet. They've always said, look, you've got to go in and you've got to biopsy the organ that's being affected. I think that's the example that we need to take. And that's where we are right now with the technology. It'd be wonderful if we could do it all in blood, but we can't. Given all of this wonderful research that's emerging, the case for biopsy becomes stronger and stronger.
Yes. And so we've got a couple of, questions, and this one is positive feedback we got. I am learning so much, ! Which is fantastic. That is why we're doing this. We want everyone to learn as we learn. But they asked, how do we get help if labs aren't showing we're sick? Any buzzwords we can use to self-advocate?
Yeah, I mean, at this point, at this point in the game, we need to really work on physician education. And we really, I say this with knowledge that it's a little bit unfortunate to have to say, but if your clinician doesn't believe that you have a complex chronic illness, sometimes the most positive thing you can do is search for a physician that will believe that you have a complex chronic illness, because right now these things are being diagnosed based on clinical findings. They're not being yet, accurately diagnosed based on blood tests. So it's just an unfortunate state of the science. This is something that it's an unfortunate state of clinical practice. The state of the science is moving forward, but again, this is our reason for being, is to accelerate the findings of the science into meaningful policy changes, meaningful lab test changes, so on and so forth. And we have the experience to do this, and we've done it in other fields. So you can bet we're going to be working hard to do it here.
Thanks. Okay, so Amy, I'd like to kind of pick your brain about some other examples of persistent pathogens and try to talk about what symptoms someone might exhibit if they may have a persistent pathogen issue.
Yeah, I mean, one of the interesting things about persistent pathogens is they could really drive a wide range of symptoms. And that is why, again, when we see patients with, let's say long COVID or other infection-associated chronic syndromes, like let's say ME/CFS or whatever, and they have a wide range of symptoms, sometimes it does help. Like if a pathogen persists, and it persists in different people in different places, it is one of the more straightforward reasons why people could have different symptoms, right? Like someone who has more persistence of a virus in their gut might have more GI symptoms, and someone who has more persistence in their lung might have more lung symptoms, right? So actually, it kind of helps explain why people develop different symptoms after an infection. But I think that one of the more interesting, that I am interested in is persistence of pathogens in nerves. And I mean, pathogens love nerves. First of all, the herpes viruses, which are players in a lot of these cases of infection associated or driven chronic illness, they are neurotrophic. And that means that they actually just primarily reside in nerves. And so if you think, they travel up and down nerves. I mean, so one of the things that interests me is that one of the most important nerves in the body is the vagus nerve. It's a nerve that innervates every major trunk organ of the body. So it innervates the gut, it innervates the heart, the lungs, and then it connects to the brain at an area of the back of the brain called the brainstem, where it then regulates via these nerve bodies in the brainstem a lot of symptoms that are really common in patients with long COVID, ME/CFS, or chronic Lyme disease, like flu-like symptoms, malaise, as part of what's called the sickness behavior response, nausea, pain, and also autonomic signaling, which is, autonomic refers to the ability kind of to move from sitting to standing, not get dizzy, like that your body's ability to regulate that. So if the vagus nerve is dysregulated, you're going to throw off the signaling of those brainstem nuclei in ways that result in some of those most common clusters of long COVID, ME/CFS, and sometimes even chronic Lyme symptoms. So one of the things that we think about a lot is that if you have a pathogen reservoir, let's say you haven't cleared the SARS-CoV-2 virus, or you haven't cleared maybe Borrelia, the Lyme pathogen, from a body site that the vagus nerve connects to, the inflammation associated with that reservoir might kind of throw off the vagus nerve in ways that result in those common sets of symptoms, even if the reservoir is kind of in different locations that vagus innervates. And the most fascinating possibility is, what if vagus is just directly infected? I just don't see why pathogens which love nerves would leave the largest nerve. That is a super highway around the body. It just helps them get from the brain to the body. Why they would leave that alone and not infect vagus. And there was an autopsy study of COVID patients, acute COVID patients, and they looked for SARS-CoV-2 RNA in the vagus nerve, and they found it in every single patient in the study. And I have to say that was interesting to me. So who knows, maybe persistence in vagus nerve itself is possible. That's actually something that Mike Van Elziger, who's my colleague at PolyBio, he wrote a paper in 2011 hypothesizing that could happen in ME/CFS. So all of these body sites could be reservoir locations, but vagus nerve sort of has extra interest to me.
And we're going to be having a dedicated education session about the vagus nerve in the coming months.
Yeah.
So if that was very interesting to you, please stay tuned for that. I am excited to learn more from you, Amy, as always.
Yeah.
And so I just wanted to remind everyone that we are taking questions. We have a couple of questions that we received from the audience that we've already answered. But if you have a question, please don't hesitate. If you're watching on YouTube, you can put it into the chat and we'll receive it. If you leave it as a comment on the YouTube video, we won't be able to see it. And if you're on Twitter or X, formerly known as Twitter, I'm also on there scanning for questions. So just wanted to remind you all as we will be concluding in 15 minutes. But yes, so let's talk about some of the future. What is the future of this field? I know that now that we have metagenomics available to us, we will be able to learn more about how to identify pathogens in biological samples. But Amy, where do you see this field moving forward? Like even in, I don't know how quickly we're progressing, but in the next year, may things even look different, or are we looking at like 5 to 10 years where we will really see a difference in how we approach pathogen persistence?
I think we're seeing an explosion of information on persistence in long COVID. We were able to sort of really get a group of people together via our long COVID research consortium. It's a group of scientists at PolyBio to study a lot of different aspects of SARS-CoV-2 persistence. So we designed a research program where we have teams working on biopsy studies and teams working on imaging studies and teams working on a range, like animal model studies, all of which are shedding light on persistence from different angles. And I think that that hasn't really been done before. And that's part of why we really honed in on this moment to be able to do that with SARS-CoV-2, because we have to be able to understand, like in order to best create diagnostics and therapies for persistence, we have to understand the mechanisms by which the pathogens persist, because they knock down parts of the immune system often to persist. They modulate mitochondrial functions sometimes as part of what they do. The more that we understand those mechanisms, the more we can create targeted ways to either disrupt those mechanisms, right? Or, additionally to find them better. So part of this mechanistic work, I think, that's happening with SARS-CoV-2 is going to be exploding. It's starting now and it will really explode over the next years. And I think we're seeing those same teams then that are interested in SARS-CoV-2 persistence in long COVID starting to pivot then into ME/CFS and the enteroviruses, which have been historically implicated often in ME/CFS outbreaks, into the tick-borne vector-borne pathogens. Like we're seeing this turning of interest. So I do think we'll see more of an explosion on that front in the coming years. The degree to which we can start to, really turn it into the clinic, it's going to be a time, but we're doing everything possible to network our way into moving it more quickly. And one thing too is, we're already running clinical trials to try to target pathogen activity, and really those trials are what's called experimental medicine, because we're running the trial and we're measuring a lot of things in trial participants so that if patients respond to, let's say we're trialing, for example, Truvada, it's an antiviral used in HIV, but Akiko Osaki, we're sending her samples and she's measuring a lot of immune and infectious and hormonal parameters in these patients. We can start to understand in people who respond to Truvada. Let's say there's a subgroup that responds well to the drug. Well, what did they have in their samples that might have differed from other people's? And that allows us in and of itself to better understand what's happening, right? David, you could probably speak more to those trials too. We're doing lumbrokinase, we're doing, a couple other ones that are kind of, experimentally medicine geared.
Yeah, no, I mean, I would start off by just saying, in terms of timeline, I do think we're looking at sort of a 5 to 10 year window rather than a 1 to 5 year window. I think that's the realistic time frame, because as Amy mentioned, we're running a lot of trials on antivirals. We're trying to initiate some trials on immunotherapies. We're running some trials on proteolytic enzymes that can affect blood vessel health, because all of these things, in this so-called invisible illness, these are things that we have, seen objective signs of dysfunction in folks with long COVID or ME/CFS or chronic Lyme disease. And, the one thing that really strikes me is is just how many unanswered questions there are right now. Right at the beginning of the pandemic, when I was very naive to post-acute infection syndromes, it had just never been part of my career. So I had no understanding of what sort of a field I was stepping into. I had heard about the theory of viral persistence, and I was asking questions. And, I was fortunate enough to be connected with some, heavy-hitting virologists who were coronavirus experts. And, I asked them directly, what about this theory of viral persistence? And they said, oh, no, it doesn't happen. And I was like, oh, cool. Can you show me papers that prove that it doesn't happen? Can you show me papers where people or animals have been infected with coronaviruses, and you've looked at their tissues and you don't see persistence? And they're like, oh, no, there aren't papers like that. We just know that it doesn't happen because, the sort of virus it is, it just doesn't stick around. And I was like, all right, that tells me everything I need to know. You learn from a textbook as opposed to people who have learned by doing, right? And people who have learned by actually being intellectually curious and looking. So enough from you. Let's talk to the viral persistence people. And then all of a sudden you start to hear, okay, well, what happens if more than one pathogen is persisting? What happens if, one pathogen is persisting plus you have POTS or dysautonomia or an autoimmune condition? So suddenly we're being flooded with questions, and and these are all worthy questions that require very systematic approaches to studying. And unfortunately, embracing the complexity is the only way that we're going to get to actionable treatments, because again, as I think both Amy and I have said multiple times, we're not going to get there with a single drug. We're not going to get there with a single blood test and then, we will give you this one medicine to fix you. We need to be turning our heads toward cancer. We need to be looking at what's happening there and the complexity that's happening there. It's not one blood test, it's multiple biopsies. Then it's sort of like, okay, you have 5 different combination treatment approaches, and each one has its own different probability based on your genetic makeup, based on your medical history. And we're going to walk you through the statistics of which one is going to be best for you in your current situation. That's the level of complexity we need to get to. And it's, again, a lot of time has been wasted, which I truly regret. I feel really, terrible about having stepped into this field and seeing how much time and how much resourcing has been wasted on things that have not helped. But, now, as Amy mentioned, there is a huge explosion of interest in the field. We need to ride that wave and get some treatments, some actionable treatments in the pipeline, so that patients can be at least getting some relief of symptoms within the next 5 years.
Thank you. I want to share this comment that we got, and they said, thank you so much. Honestly, this has been so validating as a person diagnosed with ME/CFS in 96 and EBV somewhere between that and COVID in December 2019. I've really struggled with being seen and understood by clinicians, so this gives me hope. And then they said, if you ever need trial participants, I'm ready. So do we have any information for everyone just on like clinical trials and what our status is with that?
Absolutely. Thank you to whoever made that comment. We really appreciate kind words. So we can sometimes feel a little bit, out on the edge ourselves. So it's nice to hear some words of encouragement. I would say that we are currently recruiting. At present, we have a number of clinical trials that are open to anyone who lives in New York State or within 50-mile radius of New York City, even if they're out of state. And if anyone fits that criteria, they can email CoRE Research, so CORE Research @mountsinai.org. And they can see some of the clinical trials we have going on, but also I would say check out the PolyBio Research Foundation website, because there are also a number of trials going on countrywide that can be seen.
And there are some trials, but the truth is that you would definitely have to check the geographical location of the trial. Like at the moment, it's very specific groups of patients at very specific sites. So while I hope that can expand in the future, it really is like certain targeted groups of patients right now. You basically, if you live in New York or San Francisco or Boston, there might be some options, and we hope from there we can expand a bit further in the future.
Yes, and I think you have to start somewhere, and I think it's amazing and wonderful that we have innovative people like you, David, and you, Amy, behind this, because I know you're going to drive this, as much as you can and as hard as you can to see the progress that needs to be made. And that kind of ties into one of the questions that I see here. Someone said many ME/CFS researchers slash specialists have been looking at these issues for many decades. Have you been able to reach out to them to transfer their knowledge before they retire? And I think that's a really important question. And I know that we are working on a really comprehensive suite of education and outreach initiatives coming out of the center. And we certainly have our eye and priority on provider education. But David, would you like to speak more to that?
Oh, just to say yes. Not only have we been actively learning from functional medicine doctors and neurologists and immunologists and many others who have been in this space for decades, including many who are sort of tending toward retirement and I think increasingly eager to share their knowledge, and they've been very gracious in sitting us down and speaking to us about how they've been managing ME/CFS for decades all over the world as well. So not just in the United States, but folks in Europe and Australia and other places where there are pockets of really great clinical practice for ME/CFS. But we've also, been speaking to many patients who have lived experience for decades and really listening into them about what helps them and how to navigate issues that range from, things like, medication intolerance and medication sensitivities, all the way through to treatment regimens that have been particularly effective and, new and exciting things that not even, some of the doctors who have been in clinical practice for a while have heard of. So we are casting the net wide, and we're always learning. We're doing our best to learn from everyone.
Yes, and I think that's a wonderful point to wrap up as we're heading to 3:00 Eastern. It's been a really wonderful hour with you both, and I do want to shout out, David, can I shout out tomorrow that our podcast episode is coming out? Okay, great, yes. So to that point of provider education, we've been working on a collaboration with the Reach the Line podcast, and
raise the line.
Is it raise the line?
It's fine. No.
The brain, I have my brain's not perfect. I'll say that. So the Raise the Line podcast, we've been collaborating with them on a series covering post-acute infection syndromes and interviewing providers across the spectrum of, cardiology to functional medicine to physical therapy to neurology and psychiatry and beyond, to explore all of the clinical manifestations of post-acute infection syndromes, their building blocks, it's the definition in itself, and also, talking about the community aspect to this. And just as a preview, one of the things that we've heard across the board, that I've heard across the board in these episodes, is that every single person nearly has said, listen to the patient, right? And so it's a very, very inspiring podcast series. I encourage you to listen to it. Watch our social media channels for the link to listen. And yeah, with that being said, it's 3 on the dot. So Amy and David, if there's anything that you would love to mention before we sign off, let me know before we head out.
Thank you to everyone who listened. This is the first of many, and we're excited to just sort of get the education out to the people and just keep doing what we're doing. So thank you.
Definitely.
Awesome. Thanks both for your time. We're doing this monthly, so we look forward to the next one. Bye.
Totally.
Bye, everyone.
Bye.